Hi, my name is Iago Schardong. I'm a PhD student at the Institute of Plant Breeding Genetics and Genomics at UGA, and today I'm going to show you how we screen cotton genotypes for transgene presence. Well, we need to understand what is a transgene first, and what is a gene? A gene is a stretch of DNA that has the instructions to build a functional product inside an organism, such as a protein. And a transgene is a gene that was isolated from one organism and introduced to another organism through molecular engineering techniques. And in this case, could be a bacterial gene introduced into a cotton plant. And they're usually resistance genes, such as insect, disease, or herbicide resistance. So we test against transgenes in our breeding program because we're a conventional cotton breeding program, so we don't want those transgenes. We verify seed purity because pollen doesn't respect fences. If nearby fields or breeding lines, they carry transgenes, there's this risk of cross-pollination between those plants with insects. So testing helps catching any unintended gene flow really early, and it avoids regulatory issues. A lot of these transgenes need licenses to be used, and it also avoids accidental contamination from us crossing that plant that has the transgenes with our lines and contaminating the whole progeny. To test for our transgenes, we use a testing strips, and they're called lateral flow strips. And they detect specific substances in a sample, and in this case, they're proteins produced by the transgenes in a plant. It's available for multiple crops such as corn, soybean, cotton, and other crops. And similarly, we use in human testing those lateral flow strips, such as in pregnancy tests and other rapid tests. In this case, our strip detects 5 transgenes. It has a control line to make sure that the test is working, and we have 2 insect resistance genes, a glyphosate resistance gene, which is a herbicide, a dicamba resistance gene, which is also a herbicide, and another insect resistance gene. The materials we're going to be using today are plastic tags, so we can tag our plants and locate them in the field later. 2 milliliter tubes with metal BBs. In this case, we already put the metal BBs inside the tubes. And you can also use plastic paddles to grind the tissue. We're going to use our pipette and tips, and finally our transgene testing kit, which includes a buffer, the testing strips, and a protocol. After we gather our materials, we can head back out to the crossing block, where selected genotypes for crossing are planted. Our first step is to tag our plants. The bright tag helps us identify the plant easier in the field when we come back to do the crosses. The next step is to puncture a leaf using the microtube itself to collect tissue. The sample should look like this. And we're done in the field, so let's head to the lab. With our samples collected, we'll follow the protocol provided with the kit. In this case, we'll add 500 microliters of buffer to each sample. After adding the buffer, we'll shake the tube to grind the leaf sample until it's fully homogeneous. Then, we'll add our strips and wait 10 minutes for the test results. After 10 minutes, our results show that only one of the plants tested positive for transgenes. We'll then go back to the field and cut the plant so it can't be used in the crosses. This is how we maintain our germplasm free of possible contamination in the early stages of our breeding pipeline. Thank you for watching my video on how to test plants for transgenes.